So I'm glad to see a good turnout for Chelsea's defense. It's showing everyone loves Chelsea. Um, you know, in so many ways, because Chelsea in the lab has been, just such a mature voice. I find myself always deferring to Chelsea's wisdom over and over and over, which is profound. And everyone else kind of goes to Chelsea for advice and thoughts, and also kind of conveying Chelsea's involvement. She has now 10 published papers, which is more than some assistant professors. Um, and she just had one that went online today. So she's just been a tremendous collaborator, colleague. I just have to use this term, den mother. So Chelsea also she has so many aspects of her that are great. She's a great teacher. She's a sought after mentor. And in fact, I'm really pleased and hoping she's successful in her goal of going and kind of becoming a professor in a small college. And I could just imagine the students, just all her little chicklets around her, and her guiding her chicklets. So Chelsea's projects, as you'll see, are each really superb and convey her creativity as well. Let's see. Can you mute the listeners? Yeah, thanks. So she published a review, which was really well done. She also, her first research publication from the lab, by no means her first research publication, was an idea that she came up with. And she was like, I want to use this viral protein to get rid of the pathology. And I thought to myself, right, that's going to cause lots of trouble. But never look a gift horse in the mouth. And so I was like, go for it, Chelsea. And of course, I learned to defer to her wisdom. It worked great. Okay. And then she has the other project she'll present is this really cool project, which she'll tell you about in tier one, that has been a labor of love in the lab for a while, but has really produced some really interesting and spectacular results. And Chelsea has really guided and done the whole thing kind of at each point, kind of doing these outstanding experiments that like prove a point or show something. You'll see all that too. So and so Naveed, you're here. So her next goal is to kind of move to a postdoc in Austin and deep in the heart of Texas. And from there, hopefully everything will go smoothly. So I think we're in for a real delight. I will admit, Chelsea, in her practice, went at about 2x speed. So if she goes too fast, just turn down the knob, and you can slow it down, certainly on Zoom, and or speak up. Okay, thanks. Good luck, Chelsea.
I'm just gonna check this because I can't see that it's sharing anymore.
Is your mic on?
No. It doesn't have the green screen around it. Is it still sharing? All right. Thank you. Thank you so much, Ben. That was a really nice introduction. And I want to say hi to everyone and thank those in the room and those on Zoom who took the time to be here today. I'm excited to present my research on modulating immune system activation in neurodegenerative disease. In this project, I took two different approaches to modulating immune system activation. In the first project, I manipulated expression of the RNA binding protein TIA1 in a mouse model of Alzheimer's disease and assessed TIA1's function in immune activation. And the hypothesis for this project is that TIA1 inhibits microglial activation. In the first aim, we engineered a mouse model of TIA1 conditional knockout in microglia in the P301S tauopathy mouse model of Alzheimer's. In the second project, we employed RNA sequencing to identify transcriptional changes in the P301S TIA1 conditional knockout mouse. In aim 3, we determined if microglial TIA1 has a role in mediating tau pathology. In the second project, I used an ORF57 protein from the human herpes virus 8 to interrupt the integrated stress response and reduce TDP43 pathology. The hypothesis for this project was that ORF57 inhibits the integrated stress response via PKR inhibition. And aim 1 was to overexpress ORF57 and determine the effects on oxidative stress and TDP43 pathology. Aim 2 was to determine the ORF57 network interactome in the presence of TDP43 pathology. So the title of the first project is TIA1 is required for the microglial sensome response. But before I tell you about TIA1, I'm going to introduce Alzheimer's disease. Alzheimer's disease is the most common form of dementia, affecting 1 in 9 individuals over the age of 65. Patients with Alzheimer's disease develop tau neurofibrillary tangles in the intracellular space and beta amyloid aggregates in the extracellular space. These protein aggregates are toxic to the cell and cause neuron death. In the brains of patients with Alzheimer's disease, the hippocampus is the region involved in learning and memory. And neuron death in the hippocampus in these patients with Alzheimer's disease results in memory impairments. Interestingly, less than 1% of all AD cases are caused by genetic mutations. In fact, it's 0.06%. Instead, however, there are many factors that increase your risk of developing Alzheimer's disease. And these include advanced age, family history of Alzheimer's, cardiovascular disease, education status, high cholesterol, diabetes, traumatic brain injury, and certain APOE variants. Many of these risk factors, like those highlighted in red, are involved in inflammation. And inflammation is a core characteristic of Alzheimer's disease pathology. And mutations in genes related to immune response increase the risk of developing Alzheimer's disease. So on this graph, very rare mutations that are very rare in the population can cause Alzheimer's disease, such as these in presenilin and APP. However, there are other mutations, such as these over here, that are very common in the population, but only slightly increase your risk of developing Alzheimer's disease. The genes in blue are related to immune response. And I've highlighted TREM2 here because it's an important protein in clearing beta amyloid and tau aggregation. And mutations in this protein prevent the receptor TREM2 from working properly and increase your risk of developing Alzheimer's. So chronic inflammation in Alzheimer's disease contributes to neurodegeneration. The microglia are the resident immune cells of the brain. And early in disease progression, early in disease progression, when beta amyloid starts accumulating, the microglia become activated and are beneficial. They're able to clear the beta amyloid and the cells are happy. When proteins continue to aggregate and tau neurofibrillary tangles form, Microglia become overactivated, leading to microgliosis and dysfunction. This leads to inappropriate pruning of synapses and neurodegeneration. How do microglia recognize the protein aggregates? Well, that's via the microglial sensome, which consists of microglia-specific receptors that are used to sense the environment. These proteins over here were identified via transcriptomics. And in the brain, they are almost exclusively expressed in microglia. These microglial sensome genes are receptors such as purinergic receptors, which recognize ATP from dying cells, as well as pattern recognition receptors like toll-like receptors, which recognize pathogens, and TREM2, which recognize lipids and protein aggregates. As well, the microglial sensome contains cytokine receptors, chemokine receptors, and antibody receptors. So depending upon the stimulus, microglia can assume different phenotypes. If the stimulus, for example, is lipopolysaccharide or other cytokines, the microglia can assume a pro-inflammatory phenotype and resulting in cytokine production, leading to pathogen elimination, tissue damage, and neuroinflammation. Alternatively, if the stimulus is related to neurodegeneration, such as protein aggregates, DNA damage, cell death, or lipid debris, you can get a disease-associated microglial phenotype, or a DAM phenotype, which results in upregulation of gene transcription for proteins that are involved in phagocytosis or clearance of the aggregates, including CLEX7A, CD11C, and TREM2. You may also notice difference in the morphology between the resting or homeostatic microglia and the activated microglia. Resting microglia have long branching processes and they're larger. When they become activated, their processes retract and they assume a more amoeboid, small round structure. And researchers have demonstrated that reducing inflammation actually improves tau pathology. So in a tau mouse model, researchers depleted microglia using an inhibitor, and they found that in the hippocampus, there was a decrease in pathological phosphorylated tau in the mice that had microglia depleted. They also observed this same decrease in phosphorylated tau in the cortex, indicating that inflammation and microgliosis contributes to tau pathology, and that when we reduce inflammation, we reduce tau pathology. Neurons can also contribute to neuroinflammation and protein aggregation. When neurons are stressed, they can accumulate C1q, a protein involved in the complement system. They accumulate this protein in their presynaptic terminal. Microglia can recognize this accumulation and engulf the synapse, leading to pruning and neurodegeneration. Neuroinflammation can also cause neurodegeneration. Neurons can contribute to protein aggregation through their function in stress granule formation. Stress granules are membraneless organelles that form in response to stress. And the RNA binding protein, TIA1, I told you I'd get here, contributes to stress granules and is core to tau pathology. In basal or unstressed cells, TIA1 resides predominantly in the nucleus, where it's involved in RNA splicing. When the cell becomes stressed, TIA1 translocates from the nucleus to the cytoplasm, where it interacts with other RNA binding proteins and mRNA to form stress granules and protect the contents from degradation. When the stress is removed, these stress granules disassemble. In the presence of chronic stress, these stress granules can contribute to protein aggregation. In the presence of tau pathology, these stress granules also contribute to aggregation. In basal conditions, tau, the protein, interacts with microtubules, stabilizing them. In a brain with tau pathology, the tau becomes phosphorylated and forms fibrils, leading to microtubule destabilization. This tau pathology also contributes to TO1 being depleted from the nucleus and existing in the cytoplasm, interacting with other RNA binding proteins, causing toxic stress granules and contributing to tau oligomerization. TO1 also has another role, and this is perhaps the more important or relevant role for my research, and that's its function in transcription. So TO1 consists of 3 RNA recognition motifs and a low complexity domain. Researchers have looked at the binding ability to RNA and DNA for each of these RNA recognition motifs. And they see that RNA recognition motif 1 does not bind RNA and preferentially binds DNA. However, RNA recognition motif 2 binds both RNA and DNA, indicating a role of TO1 in modulating transcription of RNA. So having told you all this, now I'm going to tell you the genesis of this project, and that has to do with the dichotomy of TO1. So in Ben's lab, researchers have been looking at the role of TO1 in tau pathology for a number of years. And what they found was that when you deplete TO1 in this tau P301S mouse, you get a reduction in tau oligomerization and a subsequent increase in fibrillar tau. This corresponds with an increase in survival, suggesting that TO1 is necessary for tau oligomerization, and knocking out TO1 is neuroprotective. However, when we look outside the central nervous system, global TO1 knockout is actually a model for rheumatoid arthritis. When you take cells from mice that do not have TO1 expressed and stimulate them with lipopolysaccharide, which is a bacterial mimic, you get increased expression of the cytokine TNF-alpha, which means that in the periphery, knocking out TO1 in immune cells increases inflammation. So this led us to the question, what is the role of TO1 on inflammation in the brain? And our hypothesis here is that conditionally knocking out TO1 in microglia of a tauopathy mouse model of Alzheimer's disease will increase inflammation and result in increased tau pathology and cell death. So the first thing we needed to check this hypothesis was a mouse model of Alzheimer's disease where we manipulated TO1 expression in the immune cells, the microglia. And to do that, we used the P301S mouse. So this overexpresses human tau with a mutation that increases its propensity for aggregation. These mice develop gliosis, synaptic loss, tau neurofibrillary tangles, changes in learning and memory, and neuronal loss by the age of about 9 months. Here's a schematic showing the loss of tissue, as well as gliosis and synapse loss. Researchers that have looked at using MRI, looked at these mice, see a loss of cortical and hippocampal volume in the P301S mice compared to the wild-type non-transgenics. But what we needed to do in these mice was specifically knock out TO1 in the microglia in order to assess its function or its role. And for this, I'd like to recognize the animal technicians in our lab who have been working on breeding these mice for 7 years. So there have been 3 of them, and they're critical to all of this research. And so for breeding these mice, we took advantage of the Cre-Lox-P system, which is taken from bacteriophage. And the Cre recombinase that we express recognizes these LOXP sequences, and in our case, excises the gene that are between the LOXP sites. So we have one mouse that has a microglial specific promoter, CX3CR1, driving Cre recombinase expression. In another mouse, we have TO1 floxed, which means it's surrounded by the LOXP sites. When we breed these two mice together, we end up with mice that express both the Cre recombinase and the TO1 flox. In cells that express CX3CR1, so microglia, the Cre is expressed, and you get excision of TO1. However, in all the other cell types where CX3CR1 is not expressed, Cre is not produced, and you retain TO1. So this breeding scheme is repeated over here. And so we took these mice and bred them with the P301S tau pathology mice, and we ended up with our transgenic knockout mice with TO1 conditional knockout and the P301S TO1 conditional knockout mice. And this is a mouse, very cute, very happy. The first thing that we needed to confirm was that TO1 was actually knocked out in the microglia. And to do that, we harvested the brains from these mice, made a single cell suspension, and then used CD11B positive magnetic beads, which bind microglia, and extracted them as well as the non-microglial flow through. We extracted the RNA, made cDNA, and did RT-qPCR. First, we looked at a microglial marker, Siglec-H, and we found that our CD11B positive fraction contained the microglia, while our non-microglial flow through did not. But perhaps more importantly, we looked at TO1 expression. And in our wild type and P301S mice, we get amplification of the TO1 gene. Meanwhile, in our TO1 conditional knockout and our P301S TO1 conditional knockout mice, there's no amplification in the microglia of the TO1 gene, indicating success. So now that we had this mouse model, we needed to figure out what was happening. So we employed RNA sequencing to identify any transcriptional changes in the P301S TO1 conditional knockout mice. And so for this project, I'd also like to recognize Sambhavi from our lab and Cheng from the Mayo Clinic for their work on the bioinformatic analysis of the bulk RNA sequencing. So this is bulk RNA sequencing from 9-month-old mice. Oops, there we go. Wild type TO1 conditional knockout, P301S, and P301S TO1 conditional knockout mice. And these are genes that are significantly upregulated in the P301S mice compared to wild type. Oh. And we find that the majority of them are involved in inflammation. And in fact, when we looked at the pathways that are upregulated in the P301S mice compared to wild type, we see that they're also involved in inflammation. So we have the adaptive immune response, mediated immune response, B cell, lymphocytes, and antigen processing and presentation. When we look at the pathways that are decreased in our P301S TO1 conditional knockout mice versus the P301S tau mice, we see decrease in the immune responsive pathways, such as cellular response to interferon gamma, antigen presentation and processing, as well as interferon beta, indicating that our P301S TO1 conditional knockout mice are not having the same immune activation as our P301S mice. And so we wanted to delve a little bit deeper, and this is when we looked at the microglial sensome. And we found that 23 of the microglial sensome genes are upregulated in the P301S mice compared to the wild type. However, none of these genes are significantly upregulated in the P301S TO1 conditional knockout mice, indicating that TO1 is required for activation of the microglial sensome. So genes that are in orange are increased in expression compared to genes that are in blue. We also looked at the disease-associated microglial genes and the complement system genes. And these genes were also upregulated in P301S mice, but not when we conditionally knock out TO1. This means that the microglia were not having transcription of genes that were related to immune activation. So we wanted to see, what do these microglia actually look like? Nope, First, we validated the results. First, we validated with RT-qPCR. And at 9 months, we see the elevation of a lot of the same genes that were upregulated in the bulk RNA sequencing in the P301S mice, but not significantly in the P301S T1 knockout mice. On this slide, we're also looking at the pro-inflammatory cytokines, interleukin-1 beta and TNF alpha. These cytokines, the expression of these cytokines was also not elevated to the same extent when we conditionally knocked out TO1, indicating that both the pro-inflammatory phenotype as well as the DAM phenotype of microglia were both inhibited. Now we wanted to see what the microglia look like. And for this part of the project, I'd also like to recognize Joanna Silva and Zita Papademetrio from the University of Portugal, Minho, who did the imaging and analysis of this blinded. So here we're looking at microglia from wild type TO1 conditional knockout mice and P301S mice. In our wild type and TO1 conditional knockout mice, we have long branching processes indicating a homeostatic phenotype. In our P301S mice, the processes are retracted and we have a more amoeboid structure indicating activation. When we conditionally knock out TO1 in these mice, we have a morphology that more resembles homeostatic than activated. And when we use metrics to quantify this cell complexity and shape, we see that these are also returning to baseline when we conditionally knock out TO1. I also looked at microglial activation. And to do that, we used a marker, CD68, which is a marker of microglial activation. So to orient you on this graph, on these slides, we have DAPI, which labels nuclei in blue, IBA1 in green, which labels microglia, and CD68, which labels activated microglia in pink. And then we have our merge. And in wild type and TO1 conditional knockout mice, there are hardly any activated microglia. In the P301S mice, however, we have about 5-fold increase in activated microglia. When we conditionally knock out TO1, the microglial activation is greatly reduced closer to baseline, indicating that TO1 is important for immune activation, and by knocking it out, we're not getting transcription of genes related to immune activation, and this is correlated with homeostatic microglial morphology. This is in contrast to the global model, TO1 knockout model of rheumatoid arthritis. So in the periphery, TO1 knockout increases inflammation. So we need to get to the bottom of that. And so we need to evaluate the differences in the inflammatory response between the brain microglia and the blood peripheral immune cells, or PBMCs. So we use lipopolysaccharide, or LPS, which is a bacterial mimic, to stimulate the immune system. And LPS binds to the toll-like receptor, and through intracellular signaling, increases NF-kappa beta and IRF3 transcription of pro-inflammatory cytokines and interferon gamma genes. These are the genes that cross the blood-brain barrier and stimulate inflammation in the brain. So to do this experiment, we took our mice and we injected them with saline, and then 3 hours later collected facial vein blood. Then we took the mice and we injected LPS, that stimulant. And 3 hours later, we collected facial vein blood and then harvested the brain. In the peripheral cells, immune cells, when we added LPS to our wild type and TO1 conditional knockout mice, we get an increase of interleukin and TNF alpha cytokine release. TO1 conditional knockout exacerbated this inflammatory response, which model or agrees with the rheumatoid arthritis model. When we look in the brain, however, there's no significant difference between our wild type and our TO1 conditional knockout mice when we add lipopolysaccharide, indicating that the microglia are somehow not responding to that increased peripheral inflammation. So we're still exploring why that is, But we're working with a couple hypotheses. One of which is that the TO1 conditional knockout and the P301S TO1 conditional knockout both have reduced macrophage infiltration in the brain. And macrophages are important in that initial LPS response. Additionally, when we look at the P301S TO1 conditional knockout mice, we see downregulation of interleukin receptors that are important for propagating the LPS response. So these are a couple of options or potential explanations that we're following up on. So now that we see that the microglia are not activated in the brain and are not responding to the tau pathology, we want to determine if microglial TO1 has a role in mediating tau pathology. And so we looked in the hippocampus, which is the region of the brain important in learning and memory. We looked in the CA3 region and the CA1 region of the hippocampus. And in Alzheimer's disease, tau assumes many different species of tau, two of which are labeled here. Thiazine red in red, which labels fibrillar tau, and MC1 in green, which labels misfolded tau. We also labeled nu N, which labels neurons, and this is a merge of all the channels. In the P301S mice, they have fibrillar tau and misfolded tau throughout the hippocampus. When we conditionally knock out TO1 in these mice, there is less tau aggregation and pathological species, indicating that reduced inflammation is reducing tau pathology. We also looked in another region of the brain, the entorhinal cortex, this is the mouse brain, entorhinal cortex, and we looked at phosphorylated tau. And in the P301S TO1 conditional knockout mouse, there's less phosphorylated tau as well. Finally, we wanted to look at oligomeric tau, which, as you remember, TO1 is involved in oligomerization of tau in neurons. But the TO1 in neurons in our mice was not impacted. Nonetheless, we did see a reduction in oligomerization and fibrillarization of the tau when we conditionally knocked out TO1 in the microglia of these mice. So, putting this all together, TO1 is necessary for activation of the microglial sensome. And TO1 conditional knockout in microglia results in less transcription of inflammatory and disease-associated marker, preservation of resting microglial morphology and reduced microgliosis, as well as decreased tau pathology. We also identified an opposing function of TO1 in the peripheral immune cells versus microglia. And just to summarize here, I made a little schematic of the microglial sensome in P301S and P301S TO1 conditional knockout mice. So in P301S mice, the microglia begin in a homeostatic state. In the presence of tau aggregates, the microglial sensome becomes activated, leading to transcription of TO1-mediated receptors, including TREM2, CLEX7A, and ITGAX. This leads to microglial activation, cytokine release, efferocytosis or clearance and pruning of dying cells, as well as increased tau pathology. When we conditionally knock out TO1 in the P301S mice, in the presence of tau pathology, microglia do not become activated, the microglial sensome does not become activated, and transcription of DAM receptors do not occur. As a result, the microglia remain in a homeostatic state and tau aggregation decreases. So the next experiments here, I focused a lot on what was happening at the cell and molecular level, and it seems that it's beneficial for the mice. But what about learning and memory? Are these actually impacting any real processes? And so behavioral testing is going to be important in the future. I also focused on the microglia, since that is what we were manipulating. However, there's a lot of other important cells in the brain, including neurons, astrocytes, and oligodendrocytes. How is TO1 affecting these cells? And we're actually following up on this project with a neuron-specific TO1 knockout model in the P301S mouse. And finally, TO1 does have a very important role in liquid-liquid phase separation and RNA granule assembly. How is that impaired in microglia? And so very quickly, or not too quickly, but now we're moving on to the second project. And that's entitled Human Herpes Virus 8 ORF57 protein is able to reduce TDP43 pathology. And we followed up with a network analysis which identifies interacting pathways. And like Ben mentioned, this was kind of a pet project of mine. And I haven't put her name on this slide, but it's in the future. I'd like to recognize Caroline Murphy, who was a master's student who took on this project and made my idea of using a viral protein to inhibit protein aggregation a reality. So thank you, Caroline, who's online right now. So to understand this project, we need to first start with viral proteins. So viral proteins have evolved to maintain protein synthesis despite activation of the integrated stress response. So when a virus infects a cell, it's trying to replicate its genome. But this host cell has antiviral processes ingrained in it that prevent this replication. And so in the presence of the viral genome, this double-stranded RNA, the integrated stress response becomes activated. And that's led by this protein, PKR, or protein kinase R. Protein kinase R recognizes the viral genome and leads to activation of the integrated stress response, specifically phosphorylation of EIF2 alpha, which is an important protein on the translation complex, leading to inhibition of translation. We also get an increase in apoptosis and production of antiviral genes. Now, the viruses have evolved to block activation of this pathway because their goal is to produce or reproduce their viral genome. And so ORF57 from human herpes virus 8 binds directly to PKR, preventing activation of this protein, which leads to EIF2 alpha not being phosphorylated and translation continuing. And just a little bit about human herpes virus 8. It's also known as Kaposi sarcoma herpes virus, discovered in 1994. And it primarily affects people who are immunocompromised and causes tumors in them. It encodes approximately 89 proteins. The one that we're most interested in is ORF57, which regulates post-transcriptional processing of viral proteins and inhibits the double-stranded RNA-dependent kinase PKR and prevents inhibition of translation. ORF57 is a viral protein, and it's very well studied because it's necessary for viral replication. It has many known interactors, including RNA binding proteins, and it's known to bind to PKR to inhibit the host antiviral response. The structure of this ORF57 resembles an RNA binding protein in that it has this intrinsically disordered region, which suggests that it's able to interact with aggregation-prone proteins. And PKR activation through this integrated stress response can contribute to TDP protein aggregation through stress granules. So during viral infection, PKR phosphorylates EIF2 alpha, which inhibits translation, but also promotes assembly of stress granules. And in these stress granules, we have TO1 and TDP43. And chronic assembly of these stress granules can contribute to TDP43 aggregation. And in fact, in 95% of sporadic ALS cases, they have TDP43 pathology. TDP43 pathology is also observed in Alzheimer's disease. Therefore, our hypothesis here is that by using ORF57 to inhibit PKR, we'll be able to prevent protein aggregation and maintain protein synthesis. So in the integrated stress response, stressors such as amyloid beta, tau monomers and oligomers, ER stress, inflammation, metabolic stress, and viral double-stranded RNA activate PKR, which leads to dimerization and phosphorylation of EIF2 alpha, leading to a decrease in global translation, an increase in stress granule assembly, and apoptosis or cell death. When we add aura 57,
Yes.
it binds directly to PKR, preventing activation of PKR and the integrated stress response.
That's why I hope she isn't even determined.
So the first thing that we needed to, I don't think they're talking to me. So the first thing that we needed to do was clone aura 57 and check whether it works and at preventing TDP43 pathology and inhibiting oxidative stress. So we used two cell lines here, the SHSY5Y, which is a human neuroblastoma cell line, and the SHSY5Y with TDP43 overexpressed in the nucleus with a green fluorescent protein, not in the nucleus, in the cytoplasm, with a green fluorescent protein tagged to it. We transduced these cells with two different constructs. Either they received an M-cherry control, which means that they'll fluoresce red, or they received ORF57 linked to an M-cherry tag. So the M-cherry will indicate that our ORF57 is being expressed. And the first thing that we needed to do was check if phosphorylation of EIF2 alpha was being affected. So we stressed these cells with oxidative stress, which increases activation of the integrated stress response and phosphorylation of EIF2 alpha.
Okay.
And we see that when we add oxidative stress, there's an increase in phosphorylation of EIF2 alpha. When we overexpress
Don't
ORF57,
need to get me coffee.
there's less, or no one's up there. That's okay. There's less expression of, less phosphorylation of EIF2 alpha. I don't know how to mute everyone from my side. Okay. So
1 second. Okay.
Yes. Yes.
it's okay. If you're on Zoom, can you mute yourselves?
You know, there we go.
I'm not muted.
You're okay.
Thanks, Ben. I have no idea.
He's muted now.
Great. Thank you.
Sure.
Oh, yeah.
Hi, floating me down.
There it is. Okay, now we're set.
Thanks,
Sorry.
Ben. So if phosphorylation of EIF2 alpha is decreased, we would also expect the integrated stress response to be inhibited, which means protein synthesis should be increased or closer to baseline, and apoptosis should be down. And when we look at protein synthesis in the stress cells, ORF57 does increase protein synthesis in the presence of stress. And when we looked at cleave caspase 3, which is a marker of apoptosis, it is also decreased when we add ORF57 compared to just M-cherry control cells. So now that we see that the Okay, so now that we see that the ORF57 is inhibiting the integrated stress response, we wanted to see what was happening to TDP43. So here we have DAPI, which labels nuclei, M-cherry, which is our construct that we're overexpressing, TDP43 in green, and G3BP1 in purple, which is a stress granule marker. When we add oxidative stress, TDP43 forms these small discrete puncta, and G3BP1 forms stress granules. When we transduce these cells with ORF57 and add the sodium arsenite stress, TDP43 forms these large amorphous granules, and stress granules decrease. We found that ORF57 increased TDP43 aggregates, both the smaller puncta and the larger aggregates. The larger granules were almost exclusively formed in the ORF57 expressing cells. We also found that TDP43 and ORF57 significantly co-localized in the presence of stress, meaning that the fluorescence signals from the TDP43 and the ORF57 overlapped. We were surprised by these results because we expected TDP43 aggregation to actually go down if the stress response was being inhibited as it appeared to be. So we wanted to follow up a little and see what these aggregates actually consisted of, since their morphology was different. So the first thing that we did was fractionation of the TDP43 into soluble and insoluble. Right here. And the first 4 lanes are our control soluble, followed by our ORF57 treated cells. And there is more soluble TDP43 and less insoluble TDP43 when we treat them with ORF57. We also looked at disassembly of our TDP43 aggregates. So for this experiment, I treated cells with sodium arsenite stress and then took it off and saw how long it took for the TDP43 aggregates to disassemble. In the M-Cherry control cells, the half-life of the TDP43 aggregates was over 2 hours. When we treated these cells with ORF57, the half-life of the TDP43 aggregates was under 1 hour. We also looked at the dynamic tendency of the TDP43 aggregates. So for this, we used FRAP, which is fluorescence recovery after photobleaching. And we take a TDP43 aggregate here in green, and we shine a laser on it to photobleach it. And depending on how long it takes for the aggregate fluorescence to recover, it indicates how dynamic or mobile the TDP43 protein is. So the more it recovers, the less is aggregated in a solid state. And when we look at the ORF57 TDP43 aggregate compared to the M-Cherry TDP43 aggregate recovery, we see that the aggregates that form those large amorphous blobs in the ORF-treated cells are less solidified and more mobile compared to the M-Cherry control TDP43 aggregates. So this was a surprising but interesting result, and we wanted to find out more what ORF57 was doing in the cell. So we did proteomics here to determine the ORF57 network interactome in the presence of TDP43 pathology. And for this project, I'd like to recognize Alejandro Ortiz and Sophie Vanderspeck from our lab, who were instrumental in doing this proteomics analysis. And for this experiment, we took our SHSY5Y cells overexpressing TDP43, and we transduced them with either ORF57 or control M-Cherry. We either added sodium arsenite oxidative stress or not, and then we lysed the cells and immunoprecipitated or pulled down the M-Cherry associated proteins and used mass spec to identify what was interacting with these proteins, with M-Cherry. And here, we're looking at the proteins on the right that are binding ORF57 and on the left that are binding M-Cherry. And I've highlighted in purple here PKR, which is a known interactor of ORF57, indicating that our proteomics is working properly. The proteins here, when we looked at the pathways that these proteins relate to, they're involved in RNA processing and translation as expected based upon the known function of ORF57. When we compare no stress to stress conditions, ORF57 appears to be binding a lot of proteins despite no pathology in the no stress cells. So when we look at the unique interactors of ORF57 in the presence of sodium arsenite stress, we come up with these 13 proteins, many of which are RNA binding proteins. In particular, and of interest to us, is TAR-DBP, which is TDP43. And here we see that ORF57 is interacting directly with TDP43 in the presence of stress, which is pretty incredible. And I'm going to give another shout out to Alejandro because he did this really cool analysis where he looked at the known interactors of TDP43 and compared them to our pull down of ORF57. And he identified 83 proteins that overlap. And many of these proteins are also involved in disease pathology, including ELAV-L1, FUS, Stoffman2, VCP, suggesting that ORF57 may be regulating multiple genes and not just the PKR pathway to affect the stress response, which is pretty cool. So in conclusion, ORF57 protects against oxidative stress in neuroblastoma cells and TDP43 aggregation cells. ORF57 also prevents insoluble aggregation of TDP43 and promotes recovery of TDP43 aggregates. And ORF57 interacts with known proteins and novel TDP43 specific proteins in a model of TDP43 aggregation. And the next steps, I put a ton of ORF57 into these cells to see what would happen. And so that's not really feasible as a gene therapy if that's where this project would go. And so it's important to titrate the expression of ORF57 where maybe we could get less off-target effects. I'd also like to look at ORF57 in a more complex model. So right now we just used SHSY5Ys, which are immortalized cell lines. And instead, it'd be nice to work in a 3D assembloid iPSC model of human cells that have multiple cell types and model human brain. And finally, as I mentioned, PKR does have other off-target effects, but there are many proteins that inhibit this integrated stress response, and we could potentially use those to inhibit PKR. And with that, I'd like to thank my funding sources, my department, Pharmacology, Physiology, and Biophysics, as well as the program in Biomolecular Pharmacology. The chairs and staff here, Vanna, Leisha, Sarah, Shelley, and David, who have been involved and were very important in all 6 years here. I'd like to thank my DAC committee for all their help and for making it work on Zoom during COVID. And then I'd like to thank my collaborators, Dr. Mo Khalil and his postdoc Giulio, for all their work on the synthetic biology project, which I didn't have time to discuss here, as well as Joanna Silva from the University of Minho and her student Zita, and Dr. Hu Li from the Mayo Clinic and his postdoc Cheng Zhang. And yes, okay. And now I'd also like to thank my lab. Over the 6 years, there have been a number of different staff in here, and we've had a lot of fun. And I just want to thank everyone for all of their support and scientific contributions and just making the lab a happy place to be in. We can all rely on each other, and that means so much in a lab. And we're always there to support each other. And I'd also like to thank Ben Wallazin, who was an amazing mentor and always pushed me and challenged me to be like a great scientist, basically. So thanks, Ben. And that's Anna with our mice and yeah, little mouse. And then I'd like to thank my friends. So we're a small cohort. It's me, Jack, and Jim. And so the PIBS program, which is much larger, adopted us in our first year. And having friends in the PhD program it makes such a difference because people know what you're going through. But also I'd like to recognize my friends outside of the lab for reminding me that there is a life outside of here. And in particular, Barbara, who came up from DC to be here. And then, of course, my cohort through the years, except for COVID. Yeah. So having them by my side has been amazing. Jack just defended. Congratulations, Jimmy's on his way, so. And then my family. So my brother and his wife, Kim, have been here. They, during COVID, when we could only interact with our pod, we played D&D. We had a great time and they kept me really sane. And then my mom and my aunt, we started working out outside and we went to Greece as well. And I'd also like to recognize my dad who passed away last year, but my parents always supported me and they never had any doubt that I would get here even when I did. And finally, I'd like to recognize Naveed, my husband, who worked in Phoenix, Arizona for 3 years and commuted back and forth so that I could stay here and do my PhD. And so thank you, Naveed. Any questions? Hi, Shelley.
Great talk, what can they say? And the talk we have lab and well. The whole team of investigators. Such important work. Of course, you know, a question of intensity. We started with TM1 having a transcriptional effect. Not only working with RNA, but working potentially with DNA and regulatory. I'd love to hear more about what you folks are. If anything is out there that people are researching to know what genes are directly affected by the.
So this is actually a very interesting finding because TIA1 is more studied in its role of translation mediation. So it binds to AU-rich elements in the 3' UTR of RNA. And so that's how it mediates translation. And so in the periphery, in those immune cells, that's how it modulates cytokine production and release. So when you knock it out, you get deregulation of those inflammatory cytokines. Now, in terms of DNA interaction and transcription, that is it's not studied as much. And in fact, there's only a few papers on its role in transcription, and they're mostly involved in DNA binding. And it seems to be that TIA1 can bind to DNA, but it can be taken off by overexpression of a polymerase, for example. And so it's unclear how TIA1 or what region of the DNA TIA1 is binding to mediate transcription and how it's affecting all of these different receptors. There are other hypotheses besides just mediating transcription, obviously. It could also mediate a pathway that's critical to activation of the immune system. So TIA1 could be having a couple of different roles, but it obviously affects transcription. Anyone else? Yes, Marcia. I don't know. That's a really good question. I would expect, yeah, I don't expect the virus to be protective in itself because it has so many other detrimental things that it's doing to the cells, besides just inhibiting the integrated stress response. So it's more like one protein could be beneficial, not the whole virus. But I'm not sure if having HHV, human herpes virus 8, affects your likelihood or not of developing ALS. That's an interesting question. Yes, David.
Almost speak like this. When I see all these elements of this interaction, and then some of it's hard to figure them out and data functions. So just wondering whether TIA1, there are a few things in TIA1 or cases of COVID, this kind of, showing that there's a effect on the glial cells, cerebral vasculature, turn affects hypertension. May not have that they do, which of course would have great consequences in the future. You see anything like that?
Well, looking at the genes, there was ACE, angiotensin converting gene, that was upregulated in P301S mice, but not in the P301S T01 conditional knockout mice that we didn't follow up on, but does relate to that question. Current therapeutics do target protein aggregation, but one of the side effects of that is the ARIA. So when the beta amyloid is cleared from the blood vessels, you get hemorrhaging or edema at those sites. And we didn't look at the role of TO1 in a beta pathology, but because TO1 affects protein aggregation, I guess it could affect beta amyloid aggregation as well. We haven't looked at that, and that could affect blood vessel and integrity. In terms of hypertension itself, I'm not sure. That's an interesting question. Yes, Pete.
Can I ask a multi-part question on the interaction of TIA and TAC?
Yes.
So I think in one of your slides you showed that when you did a conditional knockdown of microglia, you're getting less oligomer, less fibrillar TAC. And it's like discordant with the work that Dan Pico did, where we did a total knockout and we showed elevations of fibrillar TAC.
Right.
We have opinion on that. And then the second part is, have you looked at the ability of oligomeric tau to seed, to spread in the absence of TIA from the microglia?
Second question, no, but that would be really interesting to look at. And I would hope or suspect that it wouldn't spread as well if microglia are contributing to the spread, which suggests that they are, since we're decreasing the activation. The first question, so TO1 is not affected in the neurons. So that's how TO1 was being regulated in Dana Pico's work, was that TO1 was interacting directly with the tau and facilitating oligomerization. And when you knock it out, you do not get that oligomerization. When we're knocking out TO1 in the microglia, we're not affecting the oligomerization of tau in the neurons, but we're affecting the spread or the spread of the pathology, and not specifically each conformation of tau. But we don't know exactly how. Yes, Marcia, one more?
How the microglia are holding spread. Have their thoughts on how that interacts with the things of how it spreads itself to cell, possibly mediated by perhaps through the you think there's an interaction there at that level of the microglia and neurons spread the tau out from
So microglia obviously contribute to tau pathology in some function. When we deplete them, tau pathology goes down. There was a paper from Tsuneya Ikezu, who used to be a faculty here, where he showed that microglia were able to take up tau and contribute to their spread by exosome release. So microglia do have a role in spreading tau pathology. I forget the rest of your question, but.
their alternate activity between neurons. No, so that exosomal spread
Right.
between neurons is also mediated in feelings. And others show by moving for neurotransmission and activity of neurons, the changes in activity of neurons, but also with that spread. If my memory serves to be right.
Right. Right. So the microglia are also not pruning the neurons. So there's not that neurodegeneration and that release of pathological tau. So that could also be contributing to it. But the glutamate hypothesis is very relevant as well. Kelly first, yeah. Oh, I'd love to. Yeah, that would be great. Oh, I didn't. Yeah, I went with the iPSCs because I felt like that was more realistic, but I would love to put it into the mouse models. And in fact, PKR inhibition, I didn't mention this, PKR inhibition has been tested a couple different times using small drugs, small molecules, and is beneficial, but unfortunately the small molecules cause toxicity peripherally. And so there's no FDA approved. But by using a viral protein, it's more of a biological regulation of PKR activity. And so I would love to test this in mice. Yeah. Is that it? Thank you, everyone, so much. And thank you to Zoom. Thank you. I'm so sweaty. I can't actually touch you.
That's okay, that makes sense.
Oh, yeah. Everyone.
You can end the, Zoom.
Okay, let me just do a quick chat.
Yeah. Very eloquent. I don't remember that much, but it was eloquent.
No, everyone just saying congratulations.
Yeah, we didn't get time. This is the moment that, you know what I mean? Yeah.